Hello, everyone. I'm Antoine Limasset, and I'm going to present you this joint work with Camille Marché. And this is basically an advance in indexing a large collection, such as reads or genomes. And I don't think I need to convince anybody of the interest of such work. I mean, we are sitting on petabase that are sleeping in SRA because we are barely able to search it. And novel data sets came. I mean, the novel data sets are basically exploding because sequencing is becoming cheaper and more available, leading to more and more ambitious projects. So this is actually a crucial task for computational biologists to keep up to the scale of the existing and future databases. So we have to scale up our ability to index and search them. So there is many way to search a database, a way that is popular, practical, and scalable, powerful and scalable, that is actually using k-mers. So I'm really glad to be the last one on this session because you already ate a lot of k-mer. So basically, the idea is that we are going to see our sequence of interest as a set of k-mer. We are going to see our data set as set of k-mers, right? And basically, the idea is that we are going to search which data set contain, let's say, more than X interesting k-mers from our sequence of interest. And the rationale being that if a dataset contain many of your k-mer of interest, maybe the dataset is actually interesting to your question. So there is actually a very large state-of-the-art on this kind of problem. I don't have the time to cover for everything, but I advise a very nice survey here. Here, I just want to highlight the fact that there is two main family for this method: the exact method that index the k-mer in an exact way, and the inexact method that just don't. And today we are going to focus on this second part because it's usually way more scalable. And why is that so? It's because they rely on a probabilistic data structure that are very memory efficient. And most of the time, this probabilistic data structure is a Bloom filter, which is a kind of a very simple structure. Basically, you have a bit arrays, and you insert k-mer by hashing them and putting some one in the bit array. And the Bloom filter is a kind of a no maybe filter, because if it say no, you're sure that your k-mer was not inserted in the Bloom filter. But if it say yes, maybe it was your k-mer that was inserted, maybe it was another k-mer with the same hash. So, and this is what we call some false positive. But it is what is really, really nice with Bloom filter is that they are very easy to code, they are very easy to construct, but they are very, very lightweight. For example, you can put the 3 billion 31 mer of the human genome in a 8 gigabytes Bloom filter and still get a very low false positive rate. But here, that's just for one data set, and if you want to work on many data sets, we need basically one Bloom filter for each data set. And a naive way to know in which Bloom filter, in which data set your k-mer is, would be to construct n Bloom filter and to query them like separately. And it would be quite expensive, right? You have to do n query. A better way to do this is actually to build what we call an inverted index, where instead of having bit arrays encoding Bloom filters, you have bit arrays encoding slice of your Bloom filter. So one bit array will encode all the bits that appear at the given position on all your Bloom filter, which means that if you have one k-mer, you have one hash, and then you just have to perform one random access to find the answer in the presence of your k-mer in all your data set. So this method is extremely nice because it's very fast, since you only need to do one random access, which are costly. Another way to organize such Bloom filter is to do a tree. So we call this a second Bloom tree, where the leaves of the tree are the input dataset Bloom filters. And I kind of miss my laser pointer. And the internal node are the union of the sum filters. So basically, yeah, I really miss my my pointer. So this means that when you go up to the root, you have a union of more and more of your data set. So basically, the root is the union of all your input data set. And why do we do that? It's because we are able to perform a very nice query. For example, if you have a k-mer, you start by query the root. If the root say no, then you know that your k-mer is nowhere in the whole structure. So that's the end of your query. If it's in the root, then you ask on the right, you ask on the left. If the left say no, then you know that this whole part of the tree don't contain your k-mer. So you are just able to narrow down your search space each time you have a no. And if you're lucky, let's say your k-mer is specific to one dataset, you are able to make some kind of logarithmic queries, which is really nice. But one drawback of this method is that you have to create many intermediate nodes, and that you still have to do many random access and memory access when you navigate into the tree. So basically, our novel method that is called pack try to get the best from both worlds. So basically, we want to narrow down the search space while keeping the nice one random access per query. So how do we aim to do that? The first thing, which may be weird, is that we are not going to build a balanced tree like the SBT. We are going to do the opposite way. So we are going to build a super unbalanced tree. So basically, we are going to we are going to perform successive union and build this kind of what we call the comb tree, because it looks like a comb, right? And this tree is it's 8 is basically n, because we have each data set increase the 8 of this tree by 1. Okay, so why would we do that? It's because we observed that if you look at the gray comb, we call this kind of this series of Bloom filter aggregated Bloom filters, because they are all included in one another, right? So if at some point there is a 1 at a position in the Bloom filter, it goes up all the way to the root, right? So basically, if you look at a given slice, it's always 0, 0, 0, 1, 1, 1, 1, 1, 1, 1 to the end. So in fact, we can encode those slices just by encoding the position of the first one, which means that we are able to encode all those Bloom filters in the comb in log space, which means let's say we have 65,000 Bloom filters, we are able to encode them for the price of 16, which is a pretty sweet deal. So this log space comb-tree is actually very, very inexpensive, but we are still able to use it to narrow down the search space. So basically, when we perform a query, we start by looking at the comb-tree. And when it's a 2, for example, then we know that the k-mer can be found in the 2 first filters. And we know that it's nowhere after, right? So we are able to use it to narrow down the search space. But if we are unlucky, let's say the k-mer was in the first data set, then we still have to check for everywhere, right? So to mitigate this worst case, we can build another comb-tree, but using the reverse order. And this way we are able to get an information on where the k-mer can be this way and this way. And we only need to check the intersection of those two range given by the two comb-tree. So those concrete log space comb-tree are really nice. But since we already got all those input Bloom filters, and most of the query time will be spent on those, we are also able to build an inverted index on all those input Bloom filters. So we are able to narrow down the search space, but we also keep the inverted index property, that is one memory access for each query. But in fact, we can do even better. We are going to, instead of constructing a very large index, we are going to build 1,000 on very small index that are able to be treated in parallel and that can improve cache coherency. So, and we are going to split those k-mers by minimizer, which means that if we are lucky, we can get 10 successive k-mers that share the same minimizer, that will go check the same partition, and hopefully make, let's say, one random access to handle all the bunch, the bunch of k-mer. Okay, so this is pretty nice. Oh, sorry. This seems pretty nice in theory, so let's take a look at what happened in practice. So first, index construction. We see that we compared PAC with two tools of the state of the art, ODSBT and Sekotello, and we see that we were able to scale on a very large human RNA-seq collection. So we were able to go up to 32K, and I forgot to add the 64K, okay. And so, yeah, ODSBT and Sekotello were not able to scale up to PAC. And we also made some experiments on microbial collection. So the 661K from Blackwell, and more than 1 million bacterial genome assemblies from GenBank. And what's really interesting is that we were able to index such very large collection within 24 hours and using a very low amount of RAM memory, which so that our tool should be really able to scale on very, very large collection. So one drawback of the PAC method is actually the index size, because we see here that Sekotello, both Sekotello and ODSBT index are able to build a smaller index, which is kind of sad. I mean, that's the thing we want to improve next. But a very important practical point is that those tools are actually rely on very, very high temporary, very, very large temporary disk file, which means that in practice, you are not able to build those index if you don't have huge amounts of available disk, which is, once again, in practice, if we really aim to scale, it may be a problem to just build this kind of index if you don't have very, very large and efficient hard drive. So on the contrary, PAC don't use any temporary disk, so everything we write on the hard drive is the final index. And here on the microbial collection, we see that the main factors that impact the size of the index is actually the size of the Bloom filter, which means the false positive rate. So if you allow a higher false positive rate, then you are able to get a smaller index, basically. Now, the query time is where it starts to get fun, because at the beginning where we tried a batch of 100 human transcript, we basically saw that PAC was the best performing tools, but 5 minutes seemed quite expensive. And funnily, we ran the same experiment with a batch of 10,000 transcripts, and we saw that PAC and Sekotello runtime didn't change at all. And this is because it was dominated by loading the index from the disk. So the overhead was just overwhelming. And we had to go to 500,000 human transcripts to actually see the impact of the actual query, which means that we got a very, very, very high throughput, but with a kind of heavy overhead. So here of 5 minutes, but on a larger collection, it could be higher. And we also assess the impact of our logarithmic comb-tree. Is it really useful or just beautiful in theory? And we see that if we query random genome, where we expect almost no hit, because here it was Esoterica database, and we see that using 1 or 2 ABF enable the query to be almost 10 times faster. With E. coli genome, so very, very loosely related to Enterica, we still get a very nice speed up, but using other Enterica genome, we see that the speed up is not really interesting. And this is because we don't have that much negative query, so the comb-tree is not able to help us. So this is kind of expected. So this is the end of my talk. So basically, I presented PAG, that is a partitioned aggregated comb-tree technique. So yeah, we use those efficient log space comb-tree, we partition, and we still use the very nice property of the inverted index. And we are starting to work on the improved methods following the same spirit, using some strategy from find array to decrease the false positive rate. In some cases, we aim at storing approximate count. That's an application that more and more people are interested in. And we also aim to use static filters because Bloom filters are actually quite memory expensive because it's a dynamic structure. And some state-of-the-art static filters are in fact way more memory efficient and it could lead to smaller index. So thank you for your attention. If you have any question, I'm glad to answer.
Right, questions?
Thank you. Yeah, thanks for the beautiful talk. Just a very quick question. Do you have a GitHub repo or package?
Yes. Did I forget to put a link? It's a yeah, I forgot. It's a GitHub/Malfoy/pack PAC. Okay. Sorry, I just forgot. But if you have Twitter, I will redo some thread and you will find the
okay,
the link.
Thanks a lot.
Yes.
So hello, thank you for the talk. I was wondering, I think it's a question I already asked you, but some times ago, on the ordering of the data set along the comb, which I guess is very important. And did you
it's really
make
important,
any choice?
Yeah. Right now, our focus was to build an index that you could always update. Like the update is the method is exactly the method we use to construct the filter. So we didn't actually try to optimize the order of the dataset, but it would have a very, very high impact because we would be able to compress if you were able to insert similar genome together, we should be able to compress them and highly reduce the index size. But here we didn't work on this kind of problematic because we wanted to focus on an all-purpose and updatable index. But that's actually a very, very impactful things because if you insert a very, very large data set in the beginning, and if you use just one comb-tree, the comb-tree is really damaged just because it contains a lot of one very early, so you are not able to narrow efficiently the search space. So in fact, that's a very impactful statistic, but we didn't try to optimize this yet. Maybe another approach would be to say, okay, I want to index all of those data sets, like in a static way, and then perform an all versus all analysis and try to cluster the input genome and optimize the order, etc. But that's what the SBT are doing. They do some kind of pre-filtering just to decide the right way to compress their tree. But yeah, we didn't do this work yet.